Welcome to this presentation on colorectal cancer and the microbiome. Colorectal cancer is one of the most common and deadly cancers there are globally and increasing rapidly in non-elderly adults in the United States. However, there is good news. This cancer takes years to develop, so regular screening can help. Second, only about 5% is genetic in origin, so there are many proven modifiable lifestyle things you can do to reduce your risk. And lastly, the microbiome data in colorectal cancer is fascinating, growing, and indicates clear measures you can take to modify your microbiome while you still can, preferably before serious problems exist. You may ask, but how does my microbiome influence cancer? That sounds far-fetched. Actually, it is estimated, depending on the reference, some 20+% of all cancers globally originate from a virus or bacteria. It's a simple story of chronic oxidative stress, inflammation, signaling, and intracellular response driving bad protein synthesis. You're probably familiar with HPV, human papillomavirus, causing cervical cancer in women. It's the same concept, except HPV is a large family of viruses where only a handful are nefarious. Well, the same is the case in your colon. In the healthy state, you have a compartmentalized gastrointestinal microbiome, and in the colon specifically, hopefully an abundance of health-promoting bacteria, which can keep the bad actors in check. However, in colorectal cancer, not only does the lack of health promoters allow the normally present properties of pathogens to do bad things in the colon, there's an overall broken microbiome from mouth to rectum. The good news is that the microbial fingerprint for colorectal cancer is quite unique from all other conditions I've analyzed. And this uniqueness is well known in the scientific community, as many studies have focused on trying to use a microbiome report as a predictor of colorectal cancer development, and with some success. Through my comprehensive meta-analysis, I've identified this fingerprint and will explain it to you in this webinar. We're not going to cover the roughly 5% of the cancer that is genetic in origin. I can't help you change your genetic makeup. However, for the vast majority, I can help you change your genetic expression, epigenetics, which is basically turning on and off genes. We're also not going to get into the various subtypes of colorectal cancer. It's outside the scope of this presentation and not my area of expertise. We'll just keep to our understanding of this slide, that there is a slow-moving process of cellular changes that can eventually become cancerous. From a public health viewpoint, the advice given, based on the evidence, is to eat more fiber, less red and processed meat, consume less alcohol, not smoke, maintain a healthy weight, and exercise. That's great, but do people do that? The United States is number one in the world for obesity, is among the top for beef consumption per capita, and consumes about half the amount of recommended fiber. As for the world of conventional medicine, colonoscopies and surgical procedures rule the day. But what's the root cause? Perhaps if we understood that better, we could avoid a lot of expenses, morbidity, and mortality. I'm going to hit you with a lot of scientific terms to simply highlight the complexity of what's going on in the body from a biochemical viewpoint. Trust me, there won't be an example. In this diagram, we see a lot going on, but it's a pretty good representation of the evolution of CRC, colorectal cancer. Listed alongside A, we see what's called the dysbiotic microbiome. If you're watching my videos, you should be very familiar with this concept by now, as it's my number one topic. These bacteria, and others which we'll see later, are called opportunistic pathogens, another familiar term. When the environmental factors favor them, they do bad things. For example, the first bacterium listed in section A is Enterococcus faecalis, which produces enterotoxins and reactive oxygen species that cause oxidative DNA damage, induce inflammation, and damage the epithelial barrier. If we look at the list labeled B, we see the number one suspect in colorectal cancer. Ethnic nucleatum makes this thing called FADA, which enables it to adhere to and invade the cells that line your gut. It also produces a protein called FAP2, which blocks anti-tumor processes in your cells. Below this, we see the genotoxin producers. Two notables are B. fragilis and E. coli. Enterotoxin-containing B. fragilis induces tumorigenesis either by increasing the overall risk of carcinogenesis or by accelerating the process, or both. Below B. fragilis, we see what's called E. coli colobacter. Enterotoxic Escherichia coli strains, which contain what's called a PKS gene cluster, produce something called colobactin. And it's well proven that this induces double-strand breaks in mammalian DNA, a bad thing when it comes to cancer. And lastly, in D, we see that a bad diet sets the stage for these possible events. The environment, which I mentioned earlier, is largely determined by diet and antibiotic exposure, also to include PPI use. I have individual presentations on all of these topics. So what we're really talking about, which is common to so many diseases and conditions, in fact, I'd be hard-pressed to think of one that's not, is chronic oxidation and inflammation. When the health-promoting bacteria number less than they should, and the bad guys number more, the opportunistic pathogens become opportunistic, and they can do nefarious things like adhere to and invade cells. This is not a concept unique to colorectal cancer. This is a feature of opportunistic pathogens, so the same theme is shared across many diseases. Once they invade, just one example of oxidation is causing DNA breaks, which is a known precursor for cancer. The inflammation causes an environment which contributes to changes in cellular expression and therefore can be a cause or a contributor to cancer development. And we also saw the downregulation of your own anti-cancer defenses. These non-genetic cancers, known as sporadic colorectal cancer, constitute about 95% of all CRC. The three pathways by which they occur are established, and in descending order of occurrence are called the chromosomal instability pathway, CpG island methylator phenotype pathway, and the microsatellite instability pathway. The point being is what? Is that CRC is not a mystery. Once you put all the information together and you act properly, you can drastically reduce your chances of developing colorectal cancer. So, as I frequently mention, let's get to the root cause. Because the root cause is not taking a drug to reduce your inflammation, it's to get to the source of that inflammation. As far as the root cause goes for colorectal cancer, the number one bacterial suspect behind CRC is the genus Fusobacterium, which contains a number of species associated with CRC. And among those species, the chief troublemaker is F. nucleatum. The species F. nucleatum has historical data for 5 subspecies, all with a ton of ugly data. If we compile all the data at the genus, species, and subspecies level, just looking at fecal samples from my meta-analysis of 68 datasets, we get this chart. On the bottom, the x-axis, are the many major health conditions/diseases, where I've done a complete meta-analysis over the years. To the left, the x-axis, is the number of studies within each meta-analysis where a significant difference was found. Green indicates the difference was higher in healthy controls, while orange indicates it was higher in the respective disease cohort. As you can see, in comparison with all other conditions, there is a huge spike in colorectal cancer. The little stars indicate data from a meta-analysis, so one data point is actually more. In second place, we see quite a few data points for IBD, which you can learn more about in my presentations on Crohn's disease and ulcerative colitis. The point being that there is a clear association with the genus Fusobacterium and colorectal cancer, and this is just fecal data. Tumor and mucosal data tells the same story. This is just one of a number of bacteria which comprise the microbial fingerprint of CRC, which is unique. Now, within the genus Fusobacterium, we have butyrate producers. This is done through the fermentation of proteins via the lysine pathway and the glutarate pathways, both of which are from amino acid fermentation. Butyrate production is incredibly important within the GI tract, and its benefits extend to the body systemically as well. In fact, the most beneficial bacteria that we have within our GI tract, the ones that are universally or almost universally beneficial, are almost all butyrate producers. And the best of these best are butyrate producers, which ferment via the carbohydrate pathway, which you can see in orange in the diagram A and B here. They have names like Faecalibacterium faucitii, Roseburia, E. rectale, Carpococcus, and more. These are the true superheroes of the gut. However, as we established, Fusobacterium has its own butyrate production. And you might think that that would indicate that it's a health-promoting bacteria, which would be very wrong. As we saw on our previous slide, it's almost always associated with disease, And in colorectal cancer, that's particularly true. But does association equal causation? You can see here in this slide a few things. One, that Fusobacterium is a normal constituent of the oral microbiome, as evidenced by being found in 100% of the time of the oral microbiome of these subjects. Two, its high presence in colorectal cancer tissue at 57%. And three, the fact that we can prove translocation from the oral to colon microbiome by showing that 75% of the time, the detected colorectal cancer strain was identical to the oral strain from the same subject. But how does it get from the mouth to the colon? Well, it's thought that Fusobacterium translocates to the colon either by traveling down the GI tract or through the blood. As for the blood theory, there are many oral activities such as dental surgery, scaling and cleaning of the dentist, flossing and brushing at home, and even chewing, which have been associated with transient bacteremia, a spike in bacteria in the blood. In the healthy, the immune system clears these bacteria, but in the unhealthy, this extra oral bacterial burden can contribute to morbidity and mortality. The more likely route from the mouth to the colon is by traveling down the GI tract. You see, the gastrointestinal microbiome is supposed to be somewhat compartmentalized. There are certain bacteria which should ideally reside in the mouth, or the stomach, or the large intestine. When you start to see oral bacteria in the feces, it's an indication that the system is no longer self-regulating. And a bacterium which may be slightly bad in the mouth may be very bad in the large intestine, colorectal cancer being the best example of this. In the case of PPI specifically, by altering the pH of the stomach, they allow bacteria who may have otherwise not survived the acidity of normal stomach acid to pass on through, potentially causing downstream problems. As we previously established, F-nucleatum makes this thing called FADA, which enables it to adhere to and invade the cells that line your gut. This increased expression of FADA and CRC correlates with the increased expression of oncogenic and inflammatory genes. In fact, in comparison to normal tissue from healthy controls, the gut FADA level in CRC subjects is more than 10 to 100 times higher. In addition to causing grave problems in what were normal cells, F-nucleatum is very present in the eventual cancer cells arising from the parent cells, and interestingly, travels to other locations in the body if the cancer metastasizes. These higher levels of F-nucleatum in multiple studies have been correlated with lower T-cell infiltration, with advanced disease stage, higher disease recurrence, and poorer patient survival. So why is Fusobacterium so problematic in the gut? Well, it's kind of not supposed to be there. It should reside in the mouth, along with a number of other mouth bacteria which are significantly increased in the fecal microbiome of colorectal cancer patients. Mouth hygiene is very important. Not only is it a reflection of your health, it can be a direct contributor to disease. For example, there are many diseases associated with oral bacteria disseminated to a distant site as a result of transient bacteremia, and the one most people are familiar with is infective endocarditis. This is when you take antibiotics prophylactically, if you have risk factors, before invasive dental procedures. That's for bad-acting bacteria passing to distant sites via your blood, which is also possible for colorectal cancer. As we highlighted, the most likely route is passing through your GI tract. But if you can practice good oral hygiene and avoid the use of PPIs, then you're a part of the way to winning the battle of prevention. However, if you have poor oral hygiene, then these actors who are problematic for things like cavities and gingivitis can be problematic further down. Periodontal disease appears to be a bit of a group effort from the opportunistic pathogens of the mouth, and it may be a similar story in the colon, as some bacteria may play different roles in pathogenesis. In the mouth, F-nucleatum is among the pathobionts that outgrow during dysbiosis preceding periodontal disease, and it assists species such as Porphyromonas gingivalis to drive the breakdown of host microbial homeostasis and induce periodontitis. And P. gingivalis has been reported to induce oral carcinogenesis through multiple mechanisms. In fact, studies show increased P. gingivalis levels in saliva samples of patients with pancreatic and esophageal cancer. So, if it's a cancer concern in the mouth, esophagus, and pancreas, could it be a concern in colorectal cancer? Well, let's take a look at this study focused on P. gingivalis and colorectal cancer. Porphyromonas gingivalis is one of the bacteria in the microbial fingerprint for CRC. Here, the researchers looked at survival time as related to the presence and abundance of P. gingivalis in fecal and tumor samples. As seen in figures A and B, patients with fecal P. gingivalis were found to have a significantly decreased survival compared to patients without fecal P. gingivalis. Now, the number of CRC patients who actually tested positive for P. gingivalis was low, about 5%. Also, no healthy controls were positive. So again, when present, it's something certainly to take into consideration. But still, a full 95% of the CRC patients did not test positive for the bacteria. So it's a part of the puzzle. When we look at the data points for the genus Porphyromonas, which obviously includes the species P. gingivalis from the previous slides, we get this chart. This genus essentially has no data points across all major conditions/diseases I've analyzed, yet spikes through the roof in colorectal cancer. This chart includes data points for both the genus and species level. In addition, it's worse than it looks, as the asterisks below CRC represent data points for findings from meta-analyses. Bottom line, this is clearly a part of a very unique microbiome fingerprint for CRC. If you have one or more species from this genus elevated in your microbiome report, and maybe one or more from the other genera, then I would suggest it's time to take action. While no one species can be used as a reliable predictor for CRC, many studies, realizing the uniqueness of the microbial fingerprint for CRC, have attempted, and with some success, to develop equations to determine one's risk profile. Another usual suspect in CRC is the species Probiotic micro. This is another of a number of species originating from the oral cavity to cause problems in the colon for those with a dysbiotic microbiome. In both locations, in the mouth and in the gut, this bacterium can become an opportunistic pathogen, a topic I mentioned in most presentations. And it doesn't cause problems alone. The synergism it has with other oral opportunistic pathogens seems to be continued in the colon. In this study looking at saliva, fecal, subgingival fluid, and tumor tissues, P. micra, as well as two other suspects, Fusobacterium, which you now know, and Peptostreptococcus, which you'll see later, were significantly higher in the fecal samples of CRC patients as compared to healthy controls. One of the non-oral common suspects in CRC is a bacterium by the name of Bacteroides fragilis. B. fragilis is a normal member of the microbiome and has positive data as well as negative data points. But there are two types of this bacterium, one type which carries the enterotoxigenic gene and one which does not. Therefore, this study looked at mucosal levels of B. fragilis in CRC versus healthy controls to see if there was a difference in the occurrence between the two groups, and if so, could that be an effective marker to determine risk in targeted therapy. We see in advanced cancer, 100% of the CRC subjects tested positive for mucosal enterotoxigenic B. fragilis. However, 67% of the healthy controls also carried this nefarious version. This highlights the complicated world of the microbiome, and it's not an exact science. It takes an exceptionally skilled person to understand all facets. You have to understand that these healthy controls had an adequate population of health-promoting bacteria in their microbiomes to keep the toxigenic, pro-inflammatory, gene-altering subtype in check, as well as keep in check the damage it can cause. So if you find elevated levels of B. fragilis in your microbiome analysis, which by the way won't separate out the toxigenic form from the non, don't worry. We have to look at all of your results as a whole. No one bacterium is predictive all the time. Since the CRC microbial fingerprint is so unique, a number of studies have looked at the microbiota to predict risk or presence of colorectal cancer or survival after diagnosis. But as I've said for many years, although we can make a huge difference in people's lives, the world of the microbiome is still an inexact science. If we consider the number one suspect in CRC, the genus Fusobacterium, it's not present all the time. It's also present in healthy controls. We have a summary here of 3 studies highlighting presence and abundance. A carriage rate is simply if it's found at all. while the abundance is how much is found when it's detected. As you can see from this slide, Fusobacterium and other usual suspects are detected consistently, significantly, more often in colorectal subjects versus healthy controls. And when they are found, their abundance is also very frequently higher in CRC. But again, there are healthy controls who do carry these bacteria in their healthy gut microbiomes. So what makes one person more susceptible to these opportunistic pathogens? Dysbiosis, an unhealthy balance in your microbiome. There are many opportunistic pathogens who are always present in your gut, waiting for the opportunity to do bad things, hence the name opportunistic. You should prevent that from happening, and if not, your microbiome could be overrepresented by the players on our next slide. When I compile all the data for my meta-analysis, I get this slide here. The genus Fusobacterium is the clear number one suspect, and within this genus, the species F. nucleatum is highly suspect, but is certainly not alone. Additionally, the genus Porphyromonas has the second most disease-associated data points, with its species P. acrolytica leading the way within the genus, etc. on down the chart. Sure, we see here several of the usual suspects across all disease states, such as Escherichia, Enterococcus, and Streptococcus. But many of the ones on this slide are unique to CRC. When I include all biopsy data I have, which is not reflected here, it only serves to bolster these findings. In other words, these same bacteria not only are in the lumen of the colon, but also in the mucus layer and within the tumors themselves. As stated, many of these are from the oral microbiome, emphasized in previous material. My meta-analysis is enormous, but others have done smaller ones and come to the same conclusions. Let's take a look. Here we have our first of two meta-analyses of metagenomic fecal microbiome data in colorectal cancer. These researchers looked at 7 data sets, and their findings back up mine. If you look at Figure B, you see their main findings. From top to bottom are their suspects in descending order. Their number one association was F. nucleatum, while mine is the same, both at the genus and the species level. At 2 and 3, they have a couple species from the genus Probiotic, to include P. micra at number 2. I have the genus at number 4, but when just considering the species level data, I have the species P. micra at number 2 as well. They have the species Streptococcus stomatis high on the list, and I have the species high as well, and the whole genus at number 3. My number 2 genus is Porphyromonas, and they show several species from Porphyromonas on this figure to include P. acrolytica, which I also show as high on my list. On a separate note, they found higher diversity in colorectal cancer, which highlights something I've talked about for years, is that you can have very diverse, unhealthy microbiomes. So simple alpha diversity is not a good indicator of your status. Lastly, they also highlight more bacterial DNA content in the CRC groups having to do with amino acid fermentation and putrefaction, which highlights diet, overall GI efficiency, colonic pH, and toxic compound production. These are all classic findings. In our second metagenomic meta-analysis, these authors looked at 8 studies, 5 of which overlap from the previous slide. If you look at Figure A, you see many of the same names. The microbial fingerprint is unique. You don't see species from Gamella, Streptococcus, Fusobacterium, Porphyromonas, and Parvomonas this prevalent in any other disease state. This profile lends itself to creating a CRC risk score. The metagenomics data here again highlight similar findings, in that microbial genes for protein catabolism are enriched in CRC, a classic dysbiosis finding. We also see significantly enriched abundance in CRC of 2 of the virulence and toxicity factors we saw earlier, the E. coli PKS gene cluster and the F. nucleatum protein FADA. Also of interest, we see enriched mucin catabolism in the CRC genome, which brings us to the popular and interesting species called A. muciniphila. There are a number of mucin degraders in the microbiota. However, one actually has a name that fits the bill, Akkermansia muciniphila. Muciniphila meaning mucin lichen. This is a very interesting bacterium with potentially amazing benefits in IBS diarrhea, ulcerative colitis, and obesity. However, you should know that in colorectal cancer, Parkinson's disease, and the COVID-affected microbiome, Mucin is always or almost always significantly higher in the subjects with those conditions. One could make the argument that the compromised mucin layer in CRC provides more substrate or fuel for Mucin, therefore it's able to thrive. Yes, but ulcerative colitis is a classic example of a compromised mucin layer, and yet we see the opposite as shown here. To the left, we see that 100% of the time in CRC, the CRC subjects have significantly more Akkermansia muciniphila in their microbiomes than do healthy controls. Yet to the right for ulcerative colitis, 100% of the time, the healthy controls have significantly more Muciniphila in their microbiomes than do the ulcerative colitis subjects. So the mucin theory I don't think can hold water here. At the bottom, I provide you with all the PMIDs to these individual original research papers if you want to verify. This study out of India is one of those CRC PMIDs I just provided to you. We see many of the usual suspects. The green rectangles highlight the significant decreases in health promoters in CRC, like B. adolescentis, E. rectale, and F. prausnitzii, classic findings for CRC and across the board for all conditions. The orange rectangles highlight the main bad actors seen significantly higher in colorectal cancer, to include in the study. Names becoming familiar to you. The blue arrow points out Akkermansia muciniphila, which by the way is available as a probiotic. But why is a supposedly generally health-promoting bacterium so consistently high in colorectal cancer, along with other known pathogens? The truth is that we just don't know at this time. Is Muciniphila actively causing problems? Is it part of the root cause, or is it guilty by association? We can see within the orange circle the fact that Muciniphila functions perfectly fine at a higher pH. We also know that pH is higher, more basic, in dysbiosis, which includes colorectal cancer. But yet, in the other conditions where it's shown to be associated with health, those too compared to higher pH dysbiotic states. Muciniphila is also significantly higher in Parkinson's subjects and in the COVID-affected microbiome. See those presentations for more information. Therefore, in the spirit of do no harm, I do not recommend the use of this probiotic in colorectal cancer, Parkinson's disease, and in long COVID. In the green circles, we see two amazing health promoters which are negatively impacted by pH. Their counts fall off a cliff with rising pH. This is a well-proven phenomenon in studies. In general, the health promoters favor a lower or more acidic pH. Therefore, in the severely dysbiotic state, as in colorectal cancer, you still generally find their abundance significantly reduced. Here is the microbial fingerprint for the health promoters who are most consistently, significantly reduced in colorectal cancer across the 68 international human fecal microbiome datasets I included in my meta-analysis. Here we see two from the previous slide, Coccococcus and Faecalibacterium prausnitzii. We also see Bifidobacterium, a genus you're familiar with from probiotics. In other taxa to include the amazing genus Roseburia. This is a genus you've probably not heard of before, outside of my presentations, but is an incredible health promoter. It does not exist as a probiotic, but you can feed it the fuels it loves. Here we have the data points for the genus Roseburia, which as you can see across all diseases is almost entirely green, which is to say significantly higher in healthy controls as compared to cohorts with a given condition. You can see the spikes in the data for IBD, inflammatory bowel disease, and COVID here. But Roseburia is also often significantly reduced in colorectal cancer and Parkinson's disease. These bars are both a function of the number of studies available for a given condition, as well as the degree of impact within each condition. So how do we go about increasing this genus, as well as the others we saw which are significantly reduced in colorectal cancer? So what can you do to positively impact your microbiome if you have colorectal cancer concerns? Well, eating a healthy diet which includes more fiber is a good start. This paper is a meta-analysis of 21 studies just looking at that. These researchers found a significant association between higher intakes of dietary fiber and a 30% lower risk of colorectal adenoma, as shown in Figure A. Colorectal adenoma can lead to colorectal cancer. I'm not going to split hairs on the definitions of fibers and prebiotics, but what we're really looking to do is feed the good bacteria in the colon, and that's done with prebiotics. In addition, experience tells me that in a severely dysbiotic gut, which is what this is here we have in CRC, diet alone cannot drive sufficient change to correct years of damage. The average dysbiotic colorectal cancer microbiome needs properly dosed and blended prebiotics to drive environmental shift in the microbiome to one that is more health promoting. Beyond diet, are there any other measures? Sure. First, as we've seen, some oral bacteria can become problematic in the colon. Therefore, the first order of business would be better oral hygiene, which should be the norm. And this is not just brushing and flossing, but a better diet. Second would be to try to maintain the normal checks and balances within the entire GI tract. More specifically, if someone is taking PPIs, which are not medically necessary, then avoiding PPI use would be a great idea, as plenty of data shows that PPIs significantly change the microbiome for the worse. For more on this, see my presentation entitled Stomach Acid Blockers and Their Effects on the Microbiome. Lastly is, of course, to drive significant environmental shift in the microbiome, done by the intelligent use of prebiotics. My meta-analysis of the human fecal microbiome data for colorectal cancer yields the following average microbial fingerprint. The keystone taxa, which are most consistently, significantly decreased, are Bifidobacterium, Bloeschia, Coprococcus, Eubacterium hallei, Faecalibacterium prausnitzii, Roseburia, and Ruminococcus. The keystone taxa most consistently associated with colorectal cancer are Bacteroides fragilis, Enterococcus, Escherichia, Fusobacterium, Gamella, Parvomonas, Porphyromonas, Peptostreptococcus, and Streptococcus. In order to increase the abundance of the universal or almost universal health promoters within the deficient taxa, we need to feed them the fuels or substrates that they prefer. After having done a complete meta-analysis in all in vivo and humanized in vitro microbiome studies for all of the major prebiotics available in the marketplace, I have determined which prebiotics tend to feed which taxa. If you found this presentation informative, I have many more for free in my YouTube channel and also in the Microbiome University tab on my website, themicrobiomeexpert.com. There you can select from a wide variety of topics. And if you or a loved one are struggling with a disease/condition, I have condition-specific presentations as well, along with their microbiome protocols found within its respective tab on my website.